Sorry, I'm doing the setting. It takes some time.
Okay, we are live now.
Oh, great. Thank you for letting me know.
Maybe you can repeat the introduction parts.
Yeah, sure. I do it again. Sorry for the...
No worries. Thank you, Hayu.
Yeah. So hi, everyone. Welcome to the SAFARI-EFCL joint seminar. And SAFARI is a research group at ETH Zurich, led by Professor Mutulu. And the EFCL is the industry-funded research center at ETH, focused on long-term challenges in the domain of computer systems. And today we are very happy to have Professor Reetu Das talk about the landscape of genomics for system of for research. So Reetu Das is an associate professor at the University of Michigan. Prior to this, she was a research scientist at Intel Labs and research in the residence for the Center for Future Architectures Research. And some of her recent projects include in-memory architectures, custom computing for precision health and AI, fine-grained heterogeneous core architectures for mobile systems, and low-power scalable interconnects for kilo-core processors. She has received several awards, such as 2 IEEE Topics Awards and an ASF Career Award, Intel Outstanding Researcher Award, and so on. She has been inducted into IEEE ACM Micro and ISCA Hall of Fame. And feel free to start, Reetu.
Yeah, thank you, Hayu. I'm very excited to be here. Thank you for coming to listening to my talk. And hope we can have a good discussion here, not just me presenting. So would love your questions. And anything you have in mind, feel free to shoot me a question. Okay, so today I'm going to talk about systems research for genomics. Genomics is a very wide field, and we are going to just focus on hardware design for it, and how can we develop efficient hardware software co-design algorithms and architectures for it. Okay, all right. So what we have seen is that genomics, it started out very slow, and the very first human genome, which was a draft human genome, came around 2001, and it cost millions of dollars. But since then, and that was in 2001, right? So not long back, 25 years back. But since then, we have, sequencing technologies have picked up a lot of speed. What we saw was the second generation sequencing pioneered by Illumina. It was massively parallel sequencing technology. And that led to a growth spurt in how many human genomes we can sequence. And also, it also started making sequencing a part of clinical practice, not just, academic endeavor. So from the next generation sequencing technologies, which was second generation, now we even see third generation sequencing technology. So we went from Sangua to next gen to now the third gen, which is nanopore-based sequencing technology. So all this has led to a growth, and there are whole countries which are thinking to sequence all of their population soon. So this will produce massive amounts of data, and we need computing to keep up with it. So this is kind of showing the timeline of where we started with Sangua sequencing, and then to the Illumina, and which were like sort of like washing machine and refrigerator sized sequencing machines, to now handheld third generation sequencing technologies. And what's future? Well, a lot of energy and a lot of excitement with single cell sequencing, which would give even more fine grained analysis of our genetic composition, which would basically even distinguish between a cell which is from your immune system or from a separate organ, and specialize to that extent. Right, so that will again explode how much sequencing we need to do. So what we see is that sequencing technology is advancing at a breakneck space, right? I mean, $1,000 or less than $1,000 per genome is quite possible today, right? But the Moore's law, on the other hand, which kind of is a proxy of how efficient or how miniaturized hardware systems can be, is not scaling that well. So I'm preaching to the choir here. Hopefully, we have a lot of hardware folks here who understand what's the repercussions of Moore's law. So what we need is more efficient computing systems which can bridge this gap between advanced sequencing technology and slowing down hardware technology. So that's where we will do. That's where my talk is going to focus on, like, how do we bridge this gap? So before we go into system solutions, a little bit on sequencing technologies and how they have evolved, right? So things which I want to focus here on, two things, right? The throughput, right? Like how like from gigabases to terabases per day. So we started making it more and more parallel, the sequencing technology. So we have the throughput of sequencing has increased a lot. And also, the other thing which has increased a lot is like the read length. Like before, we would make these small fragments, and the earlier technologies were sequencing by synthesis, where they would have like a colonal amplification phase and then fluorescent tagging, and then you would take pictures of all these tubes, and then you would figure out what base pairs are there. Now from there, and that was pretty accurate, but the read length was short because of the structural, how much you can grow on these tubes. But from there, we have moved to nanopore-based sequencing, which is actually very fascinating. If you look under the hood, what's happening, it has like actually a biological pore with a motor protein, which can split the DNA strands, as I show here in the slide, and attract it through the pore. Now, as it passes through these tiny like biological pores, we can apply potential voltage and measure the difference, like the base pairs, depending on which base pair or cluster of base pairs which is going on, there's a difference in voltage levels, which an ADC can sense. And based on those voltage differentials, we can call the base pairs. So that's what we have moved from photosynthesis, from sequencing by synthesis by taking pictures, to kind of doing a more closed loop sensing based approach here to do the sequencing. Now, the advantage of this technology is that now you can make very long reads, like you can basically pull through almost, half to entire strands of DNA. But then because this is, there's inaccuracy in calling the base pairs, primarily because how do you distinguish between these tiny voltage potentials, especially when they're homopolymers or some very long repeated length of base pairs, and the accuracy becomes bad. So that's the trade-off here. So larger reads, which can lead to better clues in understanding our genetic composition, but at the same time, you have to overcome this inaccuracy. There are other advantages of moving to the third gen, which is other than long reads, it's small and portable. So here I'm showing a picture of like a nanopore on the field, right? So you can take it out into the field and do all your experiments and measurements. These are handheld. They can do both RNA plus DNA sequencing, which is a huge advantage if you want to understand pathogens and bugs, not just the human DNA. And it's low cost. The startup cost is very low, so it's affordable. What's interesting for hardware folks is that it has real-time and streaming output, and how we can leverage that. And I'll talk a little bit about that. So based on these, both high throughput sequencing and real-time sequencing, high throughput thanks to Illumina, and real-time thanks to Nanopore, the exploding sequencing applications. So, I mean, this we have already seen how sequencing can really help with, detecting and kind of seeing the progress of pandemic or diseases. Also in consumer genotyping, I don't know how promising that is yet, but something which I'm also more excited about is in cancer treatment. Like, you measure the progress of how well the treatment is shaping by measuring the free tumor cells. There's microbiome, which is also like holds a lot of clues to how our body works. And if we can kind of analyze that and give some diagnosis, that's very useful. There's agricultural sequencing. One thing which excites me a lot is in operating room sequencing. If we could use sequencing right during the operation, and I'll show you a project which we did for that in real time within half an hour. If we can diagnose all these variants and cancer, that would be great. And food safety, there was it's amazing if you think about, like, if you go back to the supply chain of how these big corporations work and how much food they waste because they think it has gone bad and they cannot detect a contamination early enough. So if we can do sequencing checks earlier in the supply chain, during transportation and so on, we can avoid this kind of wastage and improve sustainability. The other thing which is very exciting is liquid biopsy. What if we can, just the way we do blood tests every year for measuring our cholesterol and sugar and whatnot, can we do this annual or subannual basis to figure out if our body is developing some kind of cancer? And detecting even 2 years early is a lifetime there, right? So I think that's a very useful application of sequencing technologies. And I'll talk more about portable pathogen detection, how we can, detect a pandemic even before it starts, look at disease progression, look at superbugs, bacteria, superbugs, and so on. So sequencing,
Reetu.
yeah.
So I have a question here. So for the
Sure.
sequencing applications, because you mentioned there's many applications here, and I can imagine for some of them, for example, the agricultural sequencing, maybe you don't have a really good network in the field, right? So is there like any different requirements, or let's say the constraints for these applications when you design the hardwares? Does any concerns for that?
Yeah, no, that's a great question. So you're right, if you want to do it in the field and you don't want to send out to a lab, then you need like you need to have local edge computing there. So you need like, edge GPUs or edge ASICs, which can actually do that processing without requiring connection to the cloud. And we have some solutions in that space. So basically, edge sequencing solutions are a great poster child example for, ASICs for building domain-specific accelerators there. So yeah.
Yeah, interesting. Thank you.
Sure.
If I can have a question.
Yes, please.
Yeah, so can you please go back to the previous slide?
Yeah.
Okay, thank you very much. So basically in this picture, I see the ONT sequencer device on the space station. Is that correct?
That's right. Yeah, they're trying to use it for monitoring health of
I see.
Yeah.
So my question was, okay, which kind of applications are we going to use this device on in the space?
In the space. Yeah. So they, sometimes they want to check if they're falling sick to bugs. So maybe we can do some RNA sequencing to see how their biome composition is. That would be one way to know, if they are healthy or not. So that's one thing I can think of. The other thing is also, like, if it's a very long term, like, they're there for ages, then we can also, if they already have some kind of, progression of disease, that could be monitored there for health.
Okay, thank you. So maybe even if you have maybe astronauts are exposed to radiations, maybe that can cause some problems. And so they have. Okay, thank you very much.
Yeah, that's absolutely right. Yeah, I didn't think of that, but that sounds like a very close application which would be needed in space. Okay, great. So thank you for the questions. So we're talking about all these applications. So now let's look at a little bit about what a general precision health platform would look like. So general precision health platform, and this I'm not talking about the space applications, more like in hospitals today, I guess, would combine data sources from non-biological and biological sources, right? like electronic health records, which stores the patient history, our genomes, which is what, most of my talk will be focused at, but there's a lot more than genome. There's epigenomes, like how these genomes are transcribing. There's microbiomes in your body. Metabolomes are these small molecules which are which are floating around in your body, and how, like sugars and so on, how do they impact the health? So there's a lot more than just the genome, which could be combined. And we are very, we are very primitive in this sense. Like we have barely scratched the surface. I would say most of the precision health platforms are focused on EHR, not even including genetics. And there's some movement towards genetic, but ideal platform would use all these sources. And then it would take all these data sources and, for the biological source, we have to do some sort of sequencing, convert that analog to a digital format, right? Like that's what basically the sequencer is doing. And then with big data analytics, we will actually, do diagnosis or prevention, or even create a treatment and management plan. So this is what I would imagine 2030 precision health platforms would look like. We are not there yet, but we all have to do work to kind of do this. So this precision health platform, from our perspective as systems people, what can we do? Well, efficiency. Hayu asked a very good question about what if there's no cloud connectivity, right? So you want to reduce the form factor. That's another very important aspect to this. And we want to make it sustainable. So we want to save energy and power. We want to kind of do processing in memory, near storage, wherever it makes sense. And we want to also shrink the compute here. And then one important part of this is also security and privacy. I may not be very comfortable sharing all my biological data with a third party, right? So can we do it in a more secure and private manner? So that's another sort of a systems problem here. And it's a very interesting systems problem because this data is huge, right? Like, I mean, if you just compare, I had this comparison somewhere, like Facebook is about 0.5 gigabyte per user, and genomic data can go into like terabytes, right? So it's an interesting problem. So with that, I will switch gears to some applications which we in our group have looked over in the last 5 years and show you some examples of what kind of systems problems and solutions can be applied to this domain. So one of the first things we looked at was whole genome sequencing, and this is kind of favorite with architects, right? A lot of us work on it. But to give a very small, short preview of how this works, you get some kind of human genome sample, and then you prepare it, and then you send it through like these sequencing machines, which would basically emit reads, which are just sequences of base pairs. So these are short strings which it's emitting. Now you take these reads or short sequences and try to align it to a reference genome, which is the read alignment phase. And then you also do some quality control, like sorting, throwing away some duplicates or marking them, and so on. And after the quality control, you do variant calling. This is actually the end goal. This is the end goal, to find the differences between the reference genome and the new patient we are trying to sequence, right? So, and during the variant calling, we try to distinguish between machine errors and the variants. So machine errors are random, they're not systematic, while the variants which you will have in your genome, which are differences in your genetic code, would be systematic difference. So here in this picture, I'm showing C to A, which is a systematic difference. And so that's a variant. And then based on these variants, you can do some diagnosis. And I'll show you a small example of how, this variants can lead to diagnosis. So, so that's the kind of the pipeline we're talking about. And this is a little bit outdated data because, we used this was done probably 4 years back, I guess. But on an Amazon large CPU, it took about 14 hours to do this after we optimized the pipeline quite a bit. So we took a standard tool chain and then optimized the interfaces and all and so on. So it took about 400 CPU hours or so. Okay, so this is our starting point. Now let's see how we can do a hardware-software co-design to accelerate it. There are various steps in it, and one of the first accelerators we proposed was GenX, which tried to accelerate this read alignment's approximate string matching phase. So what we're really trying to find is these edits to the string, like, are we deleting a base pair? Are we substituting a base pair? Are we going to insert something? Are we going to do insert deletions? You know, all kinds of interesting edits can be made to the string to make the reference and read match. Classically, this is done by Bandit-Smith-Waterman. And this is order Kn. What we proposed was, instead of doing a Bandit-Smith-Waterman dynamic programming approach, we proposed a automata-based approach, starting with universal Levenshtein automata. But then we saw that, you know, the typical automata solutions, automata is just finite state machines, right? Solutions are string dependent. So now for every reference and read, you will have to program your automata to encode those strings. And we try to make it string independent by doing these retro comparison codes between the string and references. We also built a nice little systolic array and state machine to do these edits, insertion, deletion, substitutions, and figuring out the edits. So this, what we call, was SILAC's hardware accelerator. And then we did a lot of clever black FIPS to support affine gap scoring, in-place traceback, and composability. This was fabricated in 55 nanometer technology and gave a 63x speed up over a CPU solution. So that was our first accelerator which we proposed. After doing that research, we found that, you know, could we apply banding to our solution, and how can we scale it? And so that's where we realized that, you know, banding can save you a lot of redundant work. What we looked at was, like, the software is taking a full band solution, but a full band, band means, like, you know, how many sort of cells in your dynamic programming matrix are you computing the score for? We found that, you know, usually the optimal path is mostly within a narrow band. And so that can really help increase the PE utilization. But we have to make sure that in the rare cases where it is not within the band, we detect it and correct it. So we want to have optimality so that we do not miss the optimal path, but we also want area efficiency and high PE utilization. So what we ended up coming up with was a speculative solution where we would speculate that our optimal solution is within a band, and then correct our speculation in case we have missed it. Now, always in speculation, the challenge is to detect a false speculation when you made a mistake, right? So our idea there was to statically compute threshold scores, which will give you the max optimal score you can have at these edges of the band. Now, if your score is already more than that, then you are fine. You're not speculating, right? And that really worked well for this yellow region, which I'm showing you. But when we kind of cross this matrix, like, reference string is usually larger than query. So when we cross this matrix and you see this little wedge rectangle here, that's where our thresholds were not working. So for that, we said, let's do a tiny edit machine. Edit machine doesn't have a fine gap score. It's much more efficient, and it's cheaper in hardware. So can we do like a check algorithm with an edit score for this little wedge here? And if we, you know, if we are higher than the edit score, we are again good, right? So it was like a multi-level speculation, first with the thresholds and then with this edit. And with this, I mean, all this pain we took to make sure it's 100% equivalent with the software, right? Like, so it was, accuracy is very important. And I'll show you a slide actually from owner, which makes a very fun point on that. But this led to improvement of about 6x on our previous solution on an FPGA, and we had an AWS FPGA prototype for this. So that was all about doing the approximate string matching. Before you do the string matching, actually you have to find the locations. So this read, you know, it can map to different locations in the reference, and you've got to find those locations. And usually people use some kind of index solution for this. FM index is very widely used for short read sequencing. And one thing which we noted when we are kind of analyzing that part of the alignment is that this FM index is very compressed data structure. It's very small in its memory footprint. So if you look at the blue line here, it's like, and on the blue axis, FM index is about 1.5 gigabytes, right? Very compressed, very tiny. So very space efficient. But because it is so compressed, you had to do many lookups into this index to actually retrieve your data. Okay. And so memory bandwidth was becoming a bottleneck. Now, BWM-MEM2 tried to decompress it a little bit, right? And it went to about 40 gigabytes or so, but still it was very inefficient in terms of memory bandwidth. What we proposed was, what we observed was that today, servers have lots of memory, right? So why not develop a new data structure which would trade off memory capacity for memory bandwidth. So it would decrease the memory bandwidth. It will have low data requirement, but it can use more memory capacity, about 60 gigabytes or so. And we call this enumerated radix tree. So, the radix tree or suffix trees are not uncommon. They are very well-known solutions, actually. They are precursors to the FM index or Burrows-Wheeler transform. But, they become very huge. So we had to kind of make it fit within 64 gigabytes or so. So we came up with interesting, encoding schemes which would reduce the leaf nodes. It would compress the leaf nodes. It would have multi-character lookups and second index table. All this made it more like bandwidth efficient, but also storage efficient, right? So that we could actually fit it in a reasonable 64 gigabyte memory or so on. And on top of the bandwidth efficient index, we also optimized the maximal matching search. Now that will take me a lot of time to explain, but I'll just give the key insight here. Like we sort of removed a lot of redundancy between the forward and backward searches here. And that was enabled by actually the way we encoded our nodes in our index. So because of these two things, and also we had a lot of other interesting ideas in the paper, like reusing the index entries and so on. But because of these two key ideas of bandwidth efficient data structure and reducing the work with these searches, we could get a pretty good result here. What was interesting is that although, we are hardware folks, so we built the hardware accelerator for this, but even the software gave a pretty good speed up, like over a canonical software. So the algorithm and the data structure itself was more efficient, and this was integrated into the BWM-MEM2 repository, which is the de facto tool for read alignment. So this shows an example of how good code design can actually lead to improving software systems as well. And we have some people which have used it and found it useful for their practices, like actual practitioners. So now, like some plumbing work, right? So that was read alignment, which is very fascinating. I'm sure everybody loves optimizing that, but we have to do some plumbing work to make the whole pipeline work. And so we did optimize the sorting and duplicate phase, and we kind of fit it within the memory. And we brought it, like if you take out of the box, the stage is pretty slow, but we got it down to 3 minutes. After that phase comes the variant calling phase, where we're trying to find the differences after we have aligned the reads. Now, variant calling is also like a dynamic programming algorithm, where we use pair HMM is the key kernel out there. What we observed again in pair HMM, that just like our narrow band solution, a lot of these DP cells can be pruned out. And so we developed a pruning algorithm there, and then the approximate Ps were actually in the log domain. So they were very efficient, and they would help us prune. And once we have pruned, then we will do the floating point evaluation. So we used mixed precision here to kind of get efficiency as well as pruning. This was also built in an ASIC in 40 nanometer and gave us pretty good speed up with bit equivalent output, 43x speed up. So after all this, this was somewhere in 2020, I think this is a checkpoint at that time. So we kind of, with an FPGA implementation, we could get it down by about 5x. And then with an ASIC implementation, we could get it down further, 7x. So what was the bottleneck here? It was the Amdahl's law. So we accelerated a lot, but then the tiny bit, which was not very interesting to accelerate, or rather we didn't focus on it, that finally became the Amdahl's law bottleneck here. like chaining and de Bruijn assembly. So there's still, scope to kind of make that more efficient on an FPGA or on an ASIC system. So that was an exercise where we built like the whole end-to-end whole genome sequencing pipeline and hardware accelerated system for it. So this is a slide from
Sorry, can I ask a question, please?
Yes, please.
Thank you. In this other portion of the plot, how much of it would come from accessing data from storage system? Do you have an estimation of it already?
Data from where? From the disk?
Yes.
Okay. So here in the pipeline, so if you look at the blue, green, red, and like gray things which we actually accelerated, we accounted for the data movement as well. So we are kind of dividing the time based on the algorithmic steps, not necessarily based on compute and data movement. So the other would have, both compute and data together.
I see.
And whether it is compute intensive, so let's see, like I think de Bruijn graph assembly and chaining is very compute intensive. So there I won't expect a lot to be in the memory. But the graph assembly does have to access the memory more frequently. So yeah.
Okay, perfect. Thank you so much.
Sure. Great question. Okay, so accuracy matters because humans differ by a very small percentage of the genome, right? So and if you make very small mistakes also, people can be very, very different, right? So you really need to have bit equivalent and very highly accurate output, because if you read papers where it's 96% accurate, that's a human and chimpanzee, right? So that's just from my hard-earned, hard-learned journey, that please pay attention to accuracy, and the millions of dollars which are spent in making sure these software pipelines are functioning, they can diagnose diseases properly. So when we start building accelerators, pay attention to this. That's my humble request here. And we learned that the hard way from our doctor friends and our practitioners here at Michigan Medicine. So a little demo here on this was our FPGA solution.
In this demo, we show how ERT and CDEX can be used to align reads about twice as fast as software VWMM2 and call variants in the CYP2C19 gene, widely responsible for drug metabolism. After downloading the alignment outputs, we now move to the location of the CYP2C19 gene to chromosome 10. Each red block corresponds to an aligned read. We can see that more than 50% of the reads have a G to A variant in this gene.
Okay, so we did this experiment where we kind of aligned with data sets and just showing the single variant, right? So coming back to the point of accuracy. So
in this demo, we show how
let's see. Yeah. Okay, so what happens if this G2A variant is not detected correctly by accelerator, right? So this is an example, and there are many more things which can go wrong with wrong variant calls, but let's look at this one example. So this is CYP2 gene, which is responsible for drug metabolism, right? Like how do we kind of digest the drugs and how it impacts our body. So this G, when you do from DNA to RNA transcription, usually you do this GT and AG, they are the clippers, right? They help you clip the non-coding genes, and then you sort of remove the non-coding parts and create a RNA, okay? Now, what I'm showing on the right is, let's say your variant, let's say we have an A to G variant, right? So let's say the patient has this G flipped to A. So now it has this AG much further away. Instead of this first AG, we have a second AG. So this whole thing, which is actually the genetic code, would be spliced out, and there would be a 40 base pair deletion, and it would create a bad RNA. Now, because of this bad RNA, proteins won't be formed correctly, and so you won't be able to metabolize some statin drugs properly, right? So it's important, like let's say, this is an example of why, let's say a patient who is a heart patient is not reacting well to the statin drugs. And we want to get this kind of variant and give them the right answer, right? But if our accelerator is just going to make that flip, then things may not work, right? We won't be able to do this diagnosis correctly. Okay. So, so that was whole genome sequencing. Now I'm gonna show you one more application, and this was really fascinating for us to do this research. We really enjoyed doing this, and we went all the way to create a wet lab for this. So let's see what this is. So this was actually thrown as a challenge to us by our physician friends, a neurosurgeon in Michigan Medicine, Karl Koschmann. And what he told us is that when you do when you have a brain tumor, then often when we are doing the surgery, and these are very long surgeries, 12, 13-hour surgeries, we are trying to remove the tumor tissue from your brain. And to do that, what we do is we take small, very tiny, portions of the tissue and then look it up under like a very fancy microscope. That's what histologists do. And then manually inspect these aberrations and then tell you whether we are at the tumor margin or not. Okay, so that's the standard of practice now. Now, imagine it's so primitive. We are doing it with a microscope. Now, what if we could do some augment this or substitute this with molecular diagnostic? If we could take a very tiny bit of tissue, very, very tiny, and then tell you quickly within half an hour whether it's a cancer or not. That will give the neurosurgeon a much more informed decision to do and save precious tissue, right? Like we don't want to waste brain tissue, God forbid, right? So the challenge was to do this diagnosis all the way in 30 minutes. And that kind of time frame would match a histology, a frozen section histology, which is done today. So this was thrown as a blue sky challenge to us. Now, so how do we sequencing-based molecular diagnostics work? So let's look at the pipeline, how we would do it, right? So you would extract the DNA using some DNA extraction, wet lab protocol. These are very quick, 5 minutes or so. But after that, you have to do some amplification so that you can actually, because we are taking, it's precious brain tissue. So we take very tiny sample. And also the cancer cells are in less quantity, right? So they have very less amount compared to normal cells. So we want to amplify it. And then we go through a library preparation step, which is needed for your sequencer. And then you do sequencing and informatics on it, right? So what we found when we set this all up, we had an amazing postdoc, Jack, and great students who went and learned all this wet lab protocol. We got them a sequencer, and we kind of did this whole end-to-end experiment in our wet lab. What we found was that this target amplification was taking a lot of time, and this is actually a PCR step. It's in retrospective this makes sense because it's a mechanical, you have to heat and cool. So there's a thermal cycling here, which would be a lot more than just doing chemical reactions or even running the analysis on a computer. So unfortunately, we can do something about the blue thing here. We are computer scientists, right? But what do we do about this orange thing? So, it was a little bit disappointing when we saw this breakdown of time, right? So what we did was we thought, okay, let's not give up. Let's think of ways to solve this red bottleneck. And so we set up like a biosafety level 2 lab here. And this is the awesome postdoc I was talking about, Jack. Now he's actually, he transitioned to the lab of the neuroscientist I was talking about, Karl Koschmann. He's now there. He just loves this stuff. So he wants to have a career in that field. But so first thing which we tried was to co-optimize the PCR amplification time with sequencing. So we said sequencing analysis, we can do really quick and we can hide it. So can we do less amplification, but somehow do more sequencing to balance it out? And that helped us a little bit. We call this threshold sequencing. We could reduce the number of PCR cycles to some extent, right? From 30 to, let's say, 24 or so. And that got us to about 52 minutes, not enough. So what do we do now? Then what we thought of is something which I really think only a computer architect or a systems person would come up with. What we said is, we started looking at alternative target amplification techniques other than PCR. And what we saw is that some of these crazy amplification techniques, which nobody uses. And guess why? Because it is very error prone. It makes garbage output, right? So what we said is, hey, can we replace PCR with these erroneous amplification techniques, but correct it informatically? Because that's something which we know how to do. We can build algorithms for error correction. we can't do anything about thermal cycling, right? So we looked at this LAMP technology, which is also used now by Abbott and Nanopore for COVID to some extent for more faster targets. And this LAMP is very quick because it uses very complex polymerase reactions to do amplifications, right? So it's more a chemical reaction rather than thermal cycling. But it creates a lot of these folded loops, these bad products, which you need to identify correctly. And so we built like a tool called Lamprey, which would polish this and identify all these errors. And then we could do the target identification. So by doing this, we could get this green, which was the target amplification. Slowly and steadily, we got it down to, just about 20 minutes, just about 15 minutes or so, and reached our target of less than 30 minutes diagnostic. So that was, I think it was published in Communications Biology or something. But that paper is available for all of you to kind of take a look and see what was the Lamprey tool and how that can be improved. One of the challenges there is this was a single hotspot mutation from the brain bank, which we took as a tissue. And what would be great is if we can scale these techniques up to more than one hotspot mutations, have the primer design and all that, which could actually scale up. So that's an open challenge still in this work.
Okay. I have a question about this one. It's really, really interesting observation to me. Because in my mind, it's like analyzing the genome sequencing is always very challenging for the let's say computing systems. But it's like so basically you talk about several accelerators in the previous slides, and in the previous one, the computing is the bottleneck, right? And now the computing is not the bottleneck. So are they actually like different applications? So making the computing is not the bottleneck, and the computing is not the bottleneck. Is that because they are totally different applications? Is there any like common sense here?
Yes, that's absolutely right. So the previous case study was whole genome sequencing, So there it's not real time, it's actually a lab send out. You're going to the cloud, or you're doing batch processing there. Throughput is more important, compute becomes the bottleneck. But here we're trying to do it real time, at the edge, while in an operating room theater, right? So when you're trying to do it real time, you're doing only one, and you are trying to analyze one mutation at a time or two mutations at a time. We're not sequencing the whole genome, then sequencing is becoming the bottleneck. And amplification and sequencing,
I see, makes sense.
Yeah.
So it's for collaborating.
Different from different applications. That's absolutely a good observation there.
I see. Very interesting.
Sure. Okay. So the third application
as well.
Yes. Yeah.
So just a quick question. Did you manage to get the same accuracy with the Lamprey in the end? You get faster, okay, 10 minutes faster, but did you also get the same accuracy of the most accurate approach?
Yes, we did. That was the whole research for us, honestly. Everything else was in the wet lab, right? So we made sure that the Lamprey's VAF, so the variant to allele fraction, which the Lamprey came up with, was same as the PCR.
Okay.
Or within a very, very small, statistically ignored difference. Yeah.
I see. Okay, cool. Is it done all in software?
Yes, there's a GitHub. I think this is the GitHub. Yeah.
Oh yeah. Okay. Very cool.
Sure. Yeah.
Very cool. Thank you.
Thank you. Yeah.
Reetu, I also have a question.
Yes, please.
About this one. Do I remember this right, that this is used in the operation room, like in real time?
Yes.
And did I get it right that the PCR technique is just already too slow to be used in the operation room, or?
That's right.
Are there
Yeah,
uses for
it's already too slow. Yeah. So, it took about like 90 minutes after we optimized it. Yeah.
So then the only option back then was for someone to manually inspect the tissue.
Yes.
And the output of that process, did you compare that to this LAMP threshold sequencing technique in the paper? Are there any details on that?
Yeah, no, that's a great question. No, we couldn't do that. So that, because, that would require like actually samples from operating room, like from actual surgeries, which was not available to us. What we did do was we took the tissues from brain bank, right, which are like already, extracted, and they're preserving this DNA, right? And we compared the lamprey with the PCR technique. That's what we did. Now, for doing the histology analysis, which we're talking about, like for the golden truth, we'll have to actually do a clinical trial, which was very expensive, and we couldn't do that.
Okay. Thank you.
Sure.
Yeah, I guess it's very challenging to do that. Yeah.
Yeah, we need buy-in from, like, maybe Jack is doing that with Carl now.
Okay.
I should check with him. But yeah, like clinical trials can be millions of dollars, so that was beyond the scope of our tiny little lab. Yeah,
Thank you very much.
sure. Okay, so let's look at the real-time pathogen detection. So this is the third application I'm talking about. I just want to give you this flavors, right? There's so many things. So this is third, but I'm going to make it this really quick in interest of time, maybe like 3 minutes. So, I'm preaching to the choir here. We all went through COVID and we lost a lot of lives to it. And another thing which probably people don't realize, or maybe they realize, is superbugs. I mean, that can kill more than cancer. Because we see all these drug-resistant bacteria. It's already a big problem in developing countries because of bacterial misuse, because you just throw antibiotics at every small problem, and slowly we are seeing these very devilish little bacteria which can, which do not, the antibiotics just don't work on them, right? So basically we want to kind of, contain pandemics. We want to kind of make sure that we do more intelligent and smart usage of antibiotics, and we can detect superbugs if they are there in your body, so that you take a lot of prevention, right? So these are important upcoming problems, right? Now, what can we do with sequencing about this? So let me give you a COVID example. Now, when COVID started, right, do you all recall how hard it was to get the first PCR test out? I mean, we had to wait months. It was like a big deal. And why? Because PCR is actually an analog test, right? You need to build these primers, and then you need to kind of ship them out. They are like actual physical things. We have to maintain the correct temperature and so on. So there were a lot of logistical and creation problems with primer design and primer distribution. Now, what if we could have a digital primer? So, let's say what happens is, let's say, we are starting to see an emergence of a bug. Can we digitally figure out the DNA fingerprint of it? Just email each other this to all the hospitals around the world, digitally distribute it, the pathogen genome, and then do some kind of analysis to see, hey, this is happening. We are seeing a trend of pandemic. And that would be so cool, right? We don't have to depend on PCR. So having a digital primer is the key to preventing the next pandemic. And having a universal pathogen detector, something which can detect any new pathogen, anything which is coming up, right? So that's where I think sequencing can help. So advantage of this is, it's portable, it's low latency, we can detect unknown pathogens, we can instantaneously ship around the digital primers all around the world, and we can monitor the progression using strain typing and evolutionary, sorry, not evolutionary, but we can do strain typing to detect the monitor. So this is the pipeline which we propose. So we would get this DNA sample, we would sequence it, and then we would do some interpretation of what is the pathogen, what is the distribution of pathogen, and so on. So one thing which we realized here was that we could do an accelerated filter. Now, what would this filter out? So the main thing here, what we observed was that the base caller here, let me just see if my slides are, yeah. So what we observed is that the base caller is a big computational bottleneck, and also lot of the reads which we're getting out of the sequencer are actually benign human reads or host read, 99% of them. So if we can filter that out, then we can, if we build an accelerated filter to filter that out, we can really save all these sequencing time and computational time. Okay, and our interpretation will be more accurate. So we wanted to design that kind of filter. The other pretty cool thing here was that we could even what if we could design this filter to work on the voltage thresholds, on the raw squiggles? Now that would completely skip the base caller, which is basically run on, needs a GPU to run on it. It's a very large deep learning model. So we could do this read until to kind of skip the useless read, and we could also stop when we have enough data to interpret our diagnose, and we could skip the base caller to do a squiggle filter. So those were the three insights we had, and we developed an accelerator which gives, this accelerator was so tiny and so cool. It was the DTW, dynamic time warping accelerator, and it was like orders of magnitude more efficient than a CPU or GPU, just because the computation was so simple in the squiggle space. So that was the squiggle filter work, which got published 2 years back or so. Okay, so I'm gonna, give 2 minutes on a generalized solution. So far we looked at so many accelerators, right? There's a DDW accelerator, there's GenX and CDEX and so on. So let's be a bit realistic and practical here. I don't think anybody is going to build 10 different chips for one whole genome sequencing pipeline or a pathogen detection, right? So we are not going to see 20 chips. So we need like one solution which is more programmable. So think about like TPU, right? Tensor Processing Unit. It's programmable enough that it can, work on a lot of machine learning models, a lot of machine learning algorithm types, and so on. So can we have something like that for genomics? So this was our most recent work to look at, can we build such an accelerator? So we have many steps like base calling, read alignment variant calling, and different software tools. And custom accelerators can increase the performance and design cost. And there are a lot of examples here, right? Like squiggle filter from our group for base calling, GenX and GenSM from Safari. And so Darwin was from Stanford some time back, this prune pair HMM, which we developed. So there's so many point accelerators for getting this. So can we actually build like a domain-specific accelerator to bridge this gap? So we started out with benchmarking. We characterized 3 different pipelines, right? Like the whole genome sequencing, de novo assembly, and then metagenomics classification, like a pathogen detection. And after looking at these 3 different pipelines, the first thing which we observed was a lot of the kernels, time-consuming kernels in this were dynamic programming, okay? So dynamic programming was a fundamental algorithm in genome sequencing analysis. So which, if we could build, maybe it's a first step towards a generalized solution. In fact, if we look at NVIDIA, it built this DPX ISA extension. So it's a small step towards what we are thinking about. But they built some tiny CISC-like compute instruction, like max, add followed by a max and a comparator. So they build these tiny instructions, and that could, even just building few custom instructions could give them a pretty good speed up on a GPU. What we wanted to do was a bit more generalized solution for the whole dynamic programming kernel, okay? So let's take a look at what our approach was. I'll just do a quick recollection of DP programming. We initialize the, the rows, the first row and the first column, and then we slowly compute cell by cell based on previous scores, the next score. So that's how it proceeds. And so what you see is that, unfortunately, there's a dependency here, right? To compute a new cell, we need to know some previous cell. So it cannot be data parallel, arbitrarily data parallel, unless you speculate or something. And that's how we do a computation. This is a very canonical logical example. So what we looked when we analyzed all these GP kernel is that the dependency patterns can be different. Like they can be nearest neighbor, or there could be long range dependency. The other thing, what we observed was they can be 1D or 2D. So here I'm showing you matrices, but for chaining, it was just a one-dimensional DP problem. So basically the size of the matrices or the size of the vectors, the two-dimensionality, and then the dependency patterns were different. And also the precision was different. Some needed floating points, some needed just int8 was sufficient, right? So although dynamic programming was the heart of it, there were still some differences which made this a challenging problem, okay? So we wanted to make something which would take advantage of similarity, but also support all these divergences. So similarity would lead to customization, and for difference, we need to have programmability. So we looked at how to create the, we came up with this generalized framework called GenDP, which would do a kernel analysis to figure out what are the intercell and inter-PE dependencies. And then also within a cell, how do you create the objective function and map it? And then you develop an accelerator. This is what the accelerator looked like. So what you would notice here is that, we created a cluster of PE arrays. That was the common part because all the P's had nearest neighbor connections, and that would help with the local dependency. But we also had let me look. We also had custom mixed precision. So we had integer and floating point arrays that would support the mixed precision. Then we had some linkages between these clusters to support very long, one-dimensional problems, right? And then finally, let's see. Yeah, this was within a PE. So this FIFO would help to do the dependency between the extreme PEs. But in addition to that, and then we need to have a control instruction buffer and a control flow to kind of program these PEs and make sure they can handle the different dependency patterns which come with different kernels. But another interesting customization which we did, I'm trying to get to the scratchpad, but well, we'll just go with. Sorry, guys, I'm trying to rush up and finish in time. But the other interesting customization we did was we also made our control unit either configurable as 32-bit or 48-bit lanes because we had mixed precision in many of the kernels, and that configurability helped with improving the you could sequence multiple reads together. Of course, there would be divergences, but still it would increase the throughput. Then we also had this local scratchpad memory, which is how we solve the long-range dependency. This would be programmed and preloaded before you start a kernel, before you start a read, and then that would take care of those long-range dependencies, which are not nearest neighbor dependencies. So in the design takeaway, I would say that we had, for local dependency, we solved it using a one-dimensional P systolic array. The compute unit, which I didn't go into a lot of detail, was like the NVIDIA's Hopper instruction set, right? We had a reduction tree, which would capture a lot of efficient objective function calculations. That was our compute unit. And then for the differences, right? For precision, we made a mixed precision sort of pipeline there. And for dependency patterns, we looked at a FIFO structure as well as Scratchpad to kind of take care of medium to long range dependency patterns. All right. So you can look at the paper for this, but it was not as good as point accelerators. Like it cannot compete with GenX or Darwin or so on, but it was much better than a GPU or a CPU. So that's the in-between ground we're trying to reach here with the GenDP. Okay. All right. So if you're interested in this work, one starting point could be Genomics Bench. We have released this. It has 12 computationally intensive kernels, kind of covering all the works which I showed you today. So that's just a pointer here. There's a GitHub link and there's a website. You can just Google it. And none of this work can be done without my collaborators. I mean, I was very fortunate to have Satish and David, who worked with me from the systems side, and also Jenna Wiens, who's a machine learning expert. And I'm very thankful to Bob Dixon, who brought us this pathogen detection problem, and Carl Koshman, who brought to us the intraoperative neurosurgery problem, and we kind of worked with them. We learned a lot from them, not just the problems, but also the wet lab and all that stuff. So that's us. And of course, the awesome students who made all this possible. I mean, they saw no barriers. They learned microbiology, algorithms and hardware, and like they're just amazing. And most of them have graduated now, except Yufeng, I think. So yeah, thank you so much. I would stop here.
Thank you so much, Reetu. Really interesting. And I have a question about the last one that you mentioned, that you want to make a trade-off between the, let's say, the very specific accelerators and the general ones. And you find that the dynamic programming operation, it takes a long time in the all of these applications. And I found that there's another one maybe can also be dominant operations like a string matching. It can
Yeah.
so I'm just want to ask your opinion about the
Other bottleneck.
the bottleneck, also the trade-off between the ASIC, let's say the more specific accelerators and the general ones. Like, should we support more general operations to overcome the challenges to make the trade-offs between these specific accelerators and the general
Accelerator.
processors? Yeah.
Yeah, I mean, the trade-off is like, there is a large trade-off. You can look at like point solutions always, 2 to 4x better, I would say, than generalized solution. But also they are impractical, right? I don't know in a future in which we'll really have 20 chips, like for the same domain. That seems, it might happen, right? Never say never.
Yeah.
But I don't see that happening. So it's a bit more practical to develop a more generalized solution. So you do sacrifice quite a bit of performance, and you can look at the paper for those numbers, how much we do those comparisons. So that's your second question. I think the first question was that how, are there other sort of these generalized operations? I think, yeah, dynamic programming was very interesting for us algorithmically, so we took up that first. But I think the other thing which happens a lot is some kind of indexing. So there's FM index, or there's centrifuge, or there's, K-mer index. So this indexing operation is also very common. So if we can develop a common architecture for that, that would be an interesting thing to look into.
Yeah, sure.
Regarding these points, I also have a follow-up question. So you mentioned the effort into making a specialized but yet programmable and general hardware logic units for accelerating genomic applications, such as dynamic programming. So do you see also potential in making some specialized memory hierarchy for genomics?
Yeah, that's a great thought. I'm sure that's possible, and that would be very interesting because, I was talking about this indexing, and for indexing, data memory bandwidth is a big bottleneck here, right? So if you make a specialized memory hierarchy, which could trade off the memory bandwidth at different levels and, customize it, I think that would be a very useful and interesting contribution.
Like perhaps like working with k-mers as opposed to cache lines here and there.
Yeah, like instead of, spatial, cache block size definitely is not the optimal
Right.
size, right? So that's a great observation. Or, even for instance, depending on how big your index is, or how big, like, do you want to do, sometimes it might make sense to do it like in a processing in memory style, right? Like if your index is okay, it can fit in the memory and in the banks, then that's okay. Otherwise, you can even think about doing it near the storage, right? So kind of having that kind of trade-off also would be interesting.
Right. Yeah. Thanks.
Yeah, sure.
I also have a question.
Yes.
Yeah, so I was wondering if you have taken a look about the energy aspect of genomics processing or the carbon footprint emissions, and if this is also an important problem that needs to be solved.
Yeah, absolutely. I think that's a very important problem for us to solve as well. I think we are kind of taking the first steps towards it by looking at more efficient, energy efficient solutions, right? Like these custom accelerators, but there's a lot to be done there because many of my colleagues have told me that energy efficiency is not the same as sustainability. So I'm sure there's a lot of interesting opportunities there. Although I would say, the elephant in the room for that is AI. So genomics is not nearly as, as big threat to sustainability as AI. But I also see a trend that a lot of machine learning algorithms are finding their way into genomics, right? So there could be a confluence there. Like if you solve the sustainability problem for AI, you might be also solving the sustainability problem for genomics in future.
Meaning that the solutions are common.
Converging towards machine learning, for instance, base callers are today all like deep learning networks, and then variant callers are starting to migrate towards machine learning, right? So there's a lot of ML in genomics as well going forward.
Okay, I see. Okay, thanks.
Sure. All right. Well, thank you so much for listening.
Yeah, thank you so much. Is there any burning questions from Zoom and YouTube? I just checked. There's no questions from YouTube. And is there any other burning questions from Zoom audience? Okay. Okay, if there's no, let's thank our speaker again for the great talk. Thank you so much. It's really interesting. I learned a lot from your talk.
Yeah, sure thing. Thank you, Hayu, for
Yeah.
having me here. All right.
Okay, thank you. See you. Bye-bye.
Thank you for the talk.
Yeah, sure. Take care. Bye-bye.